But what I want to do today is finish up our lecture from last week on learning and memory, basic distinctions. And we talked about this very famous amnesic patient, H.M., Henry Mollison, who passed away just a couple of years ago. And he had damage to this structure lit up here, the hippocampus, but also the structure right in front of it, the amygdala. Now, I'd meant to bring in a skull to actually show you exactly how they did. The surgery which was not easy to do. I mean, just think about it for a moment. Think about how deep that is in the middle of your brain. How the heck did they get there to damage just this part of the temporal lobe? And the surgery was very, very precise. How are they actually going to do that? I think I mentioned it very, very briefly, but it's hard to get a 3D visualization unless you have actually a prop. So I brought my prop in today, my skull, and what they did with this patient and many other patients that receive this kind of surgery. Remember, I told you that this surgery is common. It's still done today. And this is surgery to relieve severe epilepsy. The big difference, what is the big difference between what they did in patient HM and what they typically do for epileptic surgery? Anybody remember? Huge difference, yes. That's right. So in HM, they took out both sides of the medial temporal lobe. And I don't, so to be precise, they took out both hippocampi and both amygdala, okay, in this patient. And that was an experimental surgery. What they still do today is only take out the hippocampus and amygdala on one side. And they determine that side to be the focus of the epileptic seizures. HM, his seizures were deemed to be so severe that they wanted to try and take out both to make sure that his seizures got better. Okay, so what they did in these patients, what they do in these patients, one approach that they use, and there are multiple, and I'm just giving you a view, is they do a craniotomy, okay? They don't do a craniotomy like this, but anyway, imagine that it's just this way. I don't have a skull that comes off less. So here, you can see the brain sitting in the skull. You remember there's a protection around the brain that we talked about. What are those called? What is the protective layers called? Meninges, okay? And so you can see there's a pretty big space between the skull and the brain, if you can see that here. And that is where this pillow of meninges filled with cerebrospinal fluid is pillowing or protecting the brain. But so let me just take the brain out. So here's the brain out of the skull. You can see your frontal lobes here are right behind your eyeballs. Frontal lobes right here. Medial temporal lobe is right down here, very low down into the temporal lobe, okay? And so what they did with patient HM is they had a craniotomy like this, exposing the frontal lobe. What they did is gently lift up on the frontal lobe so they can see the temporal lobe down here, and the surgeon went in by eye and went through the amygdala until he found this ventricle right here, and you can see the cerebrospinal fluid coming out of the ventricle. And once he found that, he knew that he was at the level of the hippocampus, and he went back with his cautery to damage both. He went in this way, lifting up this frontal lobe, damaging the amygdala, finding the hippocampus, and going back approximately 8 to 10 centimeters on both sides. So lifting up one side, peering down into the skull, and with just a little bit of an opening. Of course, he can't pull it all out. It's all connected. There's a spinal cord here connecting everything. But he can lift up the frontal lobe just a little bit to see that on both sides. It really takes a very skilled neurosurgeon to be able to do that. But that is the way that you're able to get at this very deep area of the brain. Okay, so that's some of the physical aspects. But what I really want to focus on and make sure that you understand are these kinds of patterns of spared and impaired memory functions that they observed in patient HM. Okay, so anterograde amnesia. Can somebody tell me in their own words what anterograde amnesia is? Not the definition up there, but in your own words, what does that mean? What kind of memory impairment do you see when you have anterograde amnesia? Yes? You can't learn declarative memories. Say that again? New declarative memories, perfect. You cannot learn any new declarative memories. What are declarative memories? Exactly. Declarative memories are memories that are able to be consciously recalled. So anterograde amnesia is memory impairment for the ability to learn any new declarative memories, consciously recallable memories from the time point at which you had your memory impairment or some sort of episode. This could be caused by encephalitis. The patient EP that you saw in the video last time had encephalitis, so he had anterograde amnesia from there. From that time point, people have strokes that damage their medial temporal lobe. You can have a lesion. Well, only HM had the lesion on both sides. So anterograde amnesia. HM had profound anterograde amnesia, okay? So if you asked HM to remember 5 words in a list, would he be able to learn that? No. Would he be able to remember what he did yesterday? This is after his lesion. Anything about yesterday? Would he have any memory at all about what happened yesterday? No. Okay, so think about that, the profoundness of that memory impairment. Now, retrograde amnesia. Anybody want to take a crack at what the definition of retrograde amnesia is in your own words, not the definition up here? What does that mean? I get my memory lesion, I get my hippocampal, bilateral hippocampal lesions today. Yes? That's right. I can't, say that again? That's right. If I got my lesions today, I could remember what happened before today, retrograde memory, but I can't remember anything that happens from now on. So, yes? Yes, retrograde amnesia. Amnesia is loss of memory. So retrograde memory is memory before the onset of an event. Retrograde amnesia is loss of memory for that time period. Yes, loss of memories for things that happened before you had an episode. Okay? Yes. If you have intact retrograde memory, then you have memory for things that happened before. Okay? But retrograde amnesia is loss of memory. Okay? So there's 2 terms here, retrograde memory and retrograde amnesia. Retrograde memory is memory for things that happened before your event. Retrograde amnesia means that you don't have memory for things that happened before your event. So here's your test. Does HM have retrograde amnesia? Yes or no? She said no. What do you think? Partial. He has partial retrograde amnesia because, and that's a very good point, that was the point I was about to bring up. He has partial retrograde amnesia because while he has spared memory for his childhood, he still has impaired memory for the several years leading up to his surgery. Okay? So what do you think that means in terms of what the hippocampus does? What does that mean? So we said that yes. That's right. Very long-term memories are stored elsewhere because those memories of his childhood and growing up and all his schooling are intact. Those are stored elsewhere, but what about the memories for the last few years? What? Say that one more time. They're in the hippocampus. So they may be in the hippocampus, or they may be dependent on the hippocampus. So this brings up the idea of consolidation. Consolidation in memory is the idea that memories after their forms need to be strengthened for a certain period of time before they become long-term memories. So this fits very well with the overall pattern that you see in patient HM. You absolutely need your hippocampus here to form new memories, okay? And therefore, patient HM has anterograde amnesia. He can't form any new memories, okay? You don't need your hippocampus for things that you learned a long time ago. You remember your grandmother, you remember your parents, you remember your siblings. Those are very long-term stored memories, and those aren't dependent on the hippocampus. They're not stored in the hippocampus. They are there for good, okay? Not dependent on the hippocampus. But those memories that happen and that are still being what we call strengthened or consolidated, and these are memories over the past couple of years right before his surgery, still need the strength of the hippocampus, or the presence of the hippocampus, to be able to be remembered appropriately. Yes? Okay. Yes. So if you had memory impairment and your sister was young when you got that memory impairment, would you recognize her as she got older? And the answer is no, you probably would not, unless she looks very, very similar when she was, whatever, 30 as she did when she was 5. And I know that because HM recognizes pictures of himself when he was in his 20s. But he does not recognize pictures of himself when he got to be 50 or 60 years old. This is himself. He sees himself in the mirror every day, but he has no memory of that image of himself. And he grew a beard, and he looked very different. So that's exactly analogous to your question. So you would not recognize your sister as she grew up if you only knew her as a young child, okay? Any other questions? Does everybody understand the concept of consolidation and how this spared or partial retrograde amnesia gradient, as we call it, suggests that there is a kind of a percolating, strengthening period that the hippocampus is important for. It's not only important for putting all new things in memory, but some of those things that happened a little while ago are still being strengthened, and that strengthening requires the hippocampus. Yes? Okay, that's a really good question. What is the consolidation period, and does it change as a function of your age? That is still a very controversial question. And the data right now suggests that whatever age you are, the consolidation gradient for when, if you as a person, as a human, gets hippocampal damage is still several years long. Okay, so you will have some impairment compared to your contemporary for the things that happened over the last several years. That's quite amazing, because it says that for a long, long time, the hippocampus maintains its strengthening power over these memories, and only after lots and lots of repetition and thinking back over, yes, I've seen my mother so many, many different times, that then does it become independent of the hippocampus. But there doesn't seem to be an age dependence in terms of the length of that consolidation. People argue over the absolute length and really puzzle over why it's so long, but there's no clear consensus over, there's no suggestion that there's a different length of consolidation for different ages. Any other questions about consolidation? Yes? Yeah. Right. Yeah. Yeah. Mm-hmm. Right. That's a great question. So the question is, could consolidation be based on the number of repetitions and not over time? And that is very difficult to test in amnesic patients, where you can't even be sure how many times these concepts have been repeated over the years. So can you think of a better way to test that idea? No. What about if you had a system where you had much more control? People are too messy. Can you test it some other way? Any ideas? So this is a great idea. This is a great scientific question that you've just posed. Is consolidation dependent on length of time or number of repetitions? And if I have 5 amnesia patients that come in, I can't test that because I can't control what's going on. Can you think of another way to be able to test that question? Any other way? What if you consider non-human species? Any ideas there? Yes. Great. Perfect. Yes. So she said if you have an animal, you could test it systematically and wait a while, or test it and wait a while and see if a length of time contributes to the consolidation, or take that same or another group of animals and give them lots of repetitions and see how their consolidation works. Perfect. That is the perfect example of the other major concept that we want to go over in this lecture. One is, what kinds of information can you get from studying patients with lesions? Obviously, we learned all about the memory system by studying patient HM. Really kind of opened up a whole new world, but we're getting to the point where we really need some specific answers to answer questions just like yours. And you need a better way to be able to test these things systematically. So we take an animal model system, and we can do the exact test that you said and answer that question. Okay, perfect. So that's a great lead-in. One more thing that I need to do, actually a couple more things, before we get to the value of animal model systems, okay? So we touched on this a little bit in our section on evolution, where we talked about what different animals are good for different kinds of studies. And we talked about how mice could be used for something. There's, they reproduce a lot. You can do lots of genetic studies. Rats are very good. We use monkeys in certain cases because they can perform tasks very similar to humans. And we're going to get into that. But before I go into that, I just want to spend a little time on these different dichotomies of what we've just learned based on patient HM. Patient HM, as we just went over, has impairments in what we call declarative memory. Things you know that you can tell each other. Another way to say that is things that you know that are consciously, that you're able to consciously bring to mind. That is a broad definition of declarative memory. Forms of long-term memory that are not dependent on the medial temporal lobe. So declarative memory is dependent on the medial temporal lobe. Declarative memory is what is impaired in HM. And at this point, we think it's the general medial temporal lobe area that includes hippocampus, amygdala, and maybe some other areas, just in that general region. Can't be sure. Long-term memory that is not dependent on the medial temporal lobe is called non-declarative, procedural memory. Things you know that you can show by doing. And remember, we talked about 2 tests that HM could do perfectly well but had no memory of doing those tests. What 2 tests were they? Mirror tracing is one. Backward mirror reading is another. These are just 2 examples of kind of what we call procedural types of tasks. We already did that. Okay. So I want to further differentiate declarative memory into 2 different categories, okay? 2 major categories of declarative memory are semantic memory, memory for facts, and episodic memory, Memory for events, also including autobiographical memory. These are all the memories that are really making up our own personal histories and make up our personalities. Episodic memories or event memories. What I did yesterday, the fun thing that I did this weekend, the fun thing that I'm going to do over Thanksgiving holiday. All of those are episodic memories, okay? So both of these types of memories are impaired in patient HM. He cannot learn any new facts. He cannot learn, remember any new events that had happened to him, but he can learn things that he can show you by doing, but he can't tell you that he learned them. Okay, so again, just another way to look at this long-term memory is a declarative memory includes both episodic and semantic memory, again, just emphasizing that this damage to the medial temporal lobe is impairing this form of declarative memory. Therefore, declarative memory is dependent on the medial temporal lobe, both semantic memory and episodic memory. Okay, so now we, let's just ignore this part. We just went over declarative memory. Let's talk a little bit more about non-declarative memory. Here we're looking at 3 different kinds of non-declarative memory, procedural memory. We talked about this more in detail. Skill learning, knowing how to ride a bicycle. This skill learning is the kind of mirror reading, mirror tracing that we talked about last time. Priming is a second form of memory. Priming is dependent on, typical type of priming is visual priming. It's sensory kind of, it's a sensory kind of test where you're given a picture of an image and later you're shown just a little snippet of that visual image and you're able to say that that image is familiar without even having any conscious recollection that you've seen it before. We're not going to focus so much on priming. I want to focus on skill learning and conditioning. Conditioning is a form of memory. Anybody who's taken a Psych 1 class knows about conditioning and knows about, this guy, Pavlov. Who can tell me what Pavlov did with his dogs? Yes? Yes. Right. So he taught the dog, first, what they call the unconditioned stimulus is the food. You give dog food and he salivates, okay? But then you associate the bell with the food. Every time you ring the bell, you give the food. And the dog learns to associate the bell with the food. That is conditioning. Conditioning is the association of 2 stimuli, or of a switch, or of a stimulus and a response. So Pavlov's dog experiment, and skill learning is, we already talked about, mirror drawing, mirror reading. So conditioning is the, so you can say, well, dog isn't conscious, so he can't learn this. But we have examples of conditioning that we do every single day. Time conditioning, I get really hungry at 12 o'clock every day. I don't know, it's 12 o'clock, and it's just a natural kind of conditioning. I don't salivate or anything, but it's a natural conditioning that happens. And I thought one of the best examples of everyday conditioning that that I've seen is the following little video clip. This just takes a second to load. Okay, here we go. Okay, this is conditioning you can do with your friends.
Oh, damn, lost another file. Gonna have to reboot again. Hey, Dwight, do you want an Altoid? What do you think? In school, we learned about this scientist who trained dogs to salivate at the sound of a bell by feeding them whenever a bell rang. So for the past couple weeks, I've been conducting a similar experiment. Dwight, want an Altoid? Okay. Altoid?
Sure.
Intoid?
Midbrain? Yes.
What are you doing? What?
I don't know. Well, my mouth tastes so bad all of a sudden.
Okay, so that is an excellent example of conditioning. It doesn't have to be a dog. It could be your office mate. And it really is an unconscious type of thing, that things get associated with each other. You don't even know. You do these things automatically. There's many things that we do automatically. And one of the brain structures that's critical for conditioning is the cerebellum. We're going to be talking about on Wednesday, another brain structure important for other forms of conditioning, fear conditioning, which is the amygdala. The amygdala comes in whenever fear is involved. So let's look at some of these different structures. This comes from your book, and the main thing you need to know about declarative memory, episodic semantics, is dependent on the medial temporal lobe. We're going to be talking about which structures in the medial temporal lobe are important. Non-declarative memory, skill learning, the mirror reading and mirror tracing is dependent on some of these extrapyramidal motor areas that we talked about before, basal ganglia, motor cortex, as well as the cerebellum. Priming, visual priming is underlied by visual cortex. Okay, we're not going to focus so much on that, but conditioning, you should know the basics of conditioning since it's a very, very well-known phenomenon. You should know about Pavlov's dog experiment. You should know about the office experiment that we just looked at. And this simple form of conditioning that was shown in the office video is dependent on the cerebellum. Complex conditioning, it says, involves the hippocampus. We're not going to focus on that. The other brain area that's not listed here, but again, we'll go over it on Wednesday, is the amygdala for fear conditioning. But all of that will be described on Wednesday. Okay? So what I want you to know is that for skill learning, it's basal ganglia, motor cortex, cerebellum, and for simple conditioning, Pavlovian conditioning with the salivation is also dependent on the cerebellum. Yes? More variables. A big thing is the timing between when the bell sounds and when you actually get it. Since you asked, the hippocampus is needed to bridge a longer gap, whereas the cerebellum has to have a much more precise and shorter time interval between your little bell and whatever good or bad thing is going to happen. Okay? So it gets much more complicated, but those are the basics. But you're starting to see what we call a taxonomy of memory. Okay, so imagine in 1957 when the original paper describing HM was published. We went from, remember Carl Lashley, we went from thinking that memory, it's just very complicated, it's all over the place, it's all over the cortex, we really can't study it in terms of a specific brain area, to, oh my God, this one brain area that was taken out very precisely causes profound memory impairment, but the guy's IQ was actually a little bit better than it was before he got his, before he had his surgery. Why? Because his seizures went down, so it actually helped his IQ. But he had completely normal IQ. He could solve simple memory tests. He could take the SAT and do similarly well after his surgery as he did before his surgery, because all of that information was learned before his surgery, okay? But the new thing that Brenda Milner discovered was not only better defining what was impaired, declarative memory, conscious memory for facts and events, but also identifying that in fact this was not a global memory impairment. He was able to learn certain things completely normally. That included skill learning, priming, conditioning. And that makes sense because these other brain areas that these non-declarative long-term memories are dependent on were, for the most part, spared in patient HM. The only little difference is that he did have cerebellar degeneration because the anti-epileptic drugs that he took damaged his cerebellum. But generally, he was still able to do skill learning, presumably because his basal ganglia were completely intact, and simple conditioning because his cerebellum was completely removed. It was just damaged from his anti-epileptic drugs. Okay, so starting to understand taxonomy, different kinds of memory dependent on different brain structures. And of course, this type of memory, very, very critical. Damage, the severe kind of damage that HM had. And I should say that the reason why I showed that video of EP is that EP has a similar kind of severe memory impairment as HM had. So that you would expect a similar kind of response in HM as you saw in that patient EP in the video with Alan Alda. Okay, so now we get to the question that can't be answered with studying individual human patients alone. HM was a single case study, and it was so valuable, but there's only so much we can do. We can't go in and reproduce HM. They took care of that with the paper. They said, you cannot do this anymore. This is devastating for a human to not have their declarative memory. And so what are you left to do? Well, this is where experimental neuroscience comes in, and our suggestion that was given before. How do you get at this question of what is underlying consolidation? Well, you can design experiments in what's called animal model systems. Okay, so give me some examples. Why is that so valuable? Why is that so valuable? I'm saying, okay, that's the answer. Why would that be so great to do? Yes? You can do things to animals that you can't do to humans. Yes, you can do invasive stuff, but it's more than that. Yes? Yes, you can control specifically which areas you damage, whereas if stroke patients come in, they have all these messy lesions, you can't really look at it. Great, great answer. Any other ideas why it's so useful to be able to study this in animals? Yes? Perfect. You can strictly control the environment before and after the lesion in animals in a way that you cannot do in humans. I can't take these humans and say, okay, I'm only going to let you watch or look at these kinds of things for the next 3 months that I do the study on. That just doesn't work. That's not to say you can't do some studies in humans, but gathering subjects with specific brain lesions is a very long and arduous task, and their lesions are, by definition, much more messy than you can do, you can create in an animal model situation. So fantastic. Those are exactly the answers. Those are the reasons why animal model systems are valuable. And here was the first question that we wanted to try and answer after HM was first described, way back in 1957. So they were doing these experimental surgeries. Scoville, Dr. Scoville, the neurosurgeon, was not interested in trying to identify brain areas important for memory. He was just trying to help his patient. So the study wasn't done in any experimental way. We certainly got a lot of data out of it. But the first question was, you damaged these medial temporal lobe structures. Which of these structures, when damaged, was really causing the severe anterograde memory impairment seen in patient HM and partial retrograde memory impairment? There was a general region in there. Which structures were critical? We already know the hippocampus and the amygdala were damaged, okay? And you can't study it in patients because there's so few. There's only one HM. And EP came along, 40 years later. And that was just a lucky, not for him, but it was encephalitis that happened to be quite specific on both sides, very, very rare. And for these questions, we turn to animal model systems. So turn to an animal model. What kind of animal do you want to use, and what kind of task do you want to test? memory on. You want it to be able, you want the animal to be able to do a similar kind of memory that we know is impaired in humans, that HM, that is a kind of memory that HM is impaired at. So we certainly can't go up and ask the monkey, hey, what'd you have for breakfast yesterday, all right? He can't answer those kinds of questions, even though I've been telling you that's a quintessential form of memory that is impaired with medial temporal lobe lesions. So what they did is they chose a relatively simple memory task that HM was impaired at and that animals can do. They started with animal models in monkeys, okay? And so here is the task that animals were asked to do. So first I'm going to show you different things. Have you seen this before in class? Have I showed you this before? No. Good recognition memory. Have I showed you this before? No. Have you seen this before? Yes. Okay. Have you seen this before? No. Okay. So it's a simple recognition memory task. And after a certain delay interval, HM cannot remember whether he's seen it before. Okay? This is simple recognition. He doesn't even have to name, you know, notepad and hand cream and tape measure. But he has a terrible, what they call, recognition memory. So this seemed to be a very simple thing that you could ask animals to do. And so what they developed was a task called delayed non-matching to sample. It's a very complicated name for a very simple task. It is a recognition memory task based on that task that I just showed you. Now, you have to be able to motivate the animals to choose different objects, and turns out animals love raisins. Monkeys love raisins. And so what happens is animals are in a testing situation like this. They're sitting in a little testing cage, and they're sitting in front of a platform, usually with 3 holes in it. And what happens is, when they can't see what's going on, I place an object over the central well, and underneath that well is a raisin, okay? And I raise the animal's door, so he has perfect access to it, and he sees this object, he displaces it to get his raisin, okay? But he sees the object, he must touch it, he must displace it to get his raisin. And then I close off his access. There's actually a door that comes on, he can't see it anymore. And then after a short delay interval, what I do is I show him the same object and a new object, now over the lateral, the 2 side little wells. Only one of these objects has a raisin underneath it, and that is the non-matching or the new object, okay? And so he learns that if he displaces the object that he did not see on that sample trial before, he will get a raisin. If he displaces this one, he sees nothing, and I close the door before he runs over and tries to grab this, because he's a monkey and he's very clever. They're really fast, too, so you have to open and close the doors very quickly. And so that's what you see him doing here. Here is the sample object. Where's my little thing? Here's the sample object. It's the key. He gets a raisin underneath that key. And then he sees the key and this blue cup, and he opens up the cup, and there is a raisin underneath here. There's no raisin underneath there. So it's called delayed non-matching to sample because he chooses the non-matching stimulus to the sample shown at the beginning of the trial. The other reason we use a non-matching to sample, or we ask the animal to choose the novel stimulus, is that monkeys have attraction to novelty. They would rather go to the novel stimulus anyway. They're curious. And so it's actually easier for them to learn. And so what we can do is teach them the principle of you have to choose the novel stimulus with a very short delay between the sample and the test. But then we could tap their memory capacity by showing them the sample and then giving them 30 seconds, 90 seconds, 3 minutes, and 10 minutes before we show them the actual test. And that will start to tap their more long-term recognition memory function, okay? So this was a very, very, what we call a benchmark task to study memory function in monkeys. It was chosen to be a task. The same task is impaired in patient HM. We can do it in a similar way in monkeys, although we tend to use shorter delay intervals. Monkeys don't have quite as long a memory span, I mean normal monkeys, compared to humans, but a very, very similar task. So what happens here? So the person that is credited with really doing this the right way for the first time in an animal model system to ask the question, what are the brain structures underlying declarative memory that are impaired in patient HM, was Mort Mishkin. He published a paper in the very prestigious journal Nature in 1978. And what he did is he basically reproduced the lesion in patient HM in monkeys using a similar approach to damage these structures, but then he tested two specific hypotheses. What were these hypotheses? So here's another schematic of the delayed matched samples task. You see an object, you displace it, you get a plus means as a reward underneath it. After a variable delay, you give the same object a novel object, and the plus is now underneath the novel object. We use junk objects to test these monkeys. And you can look at the performance. Here is percent correct performance on performance of this simple recognition memory task with a 30-second delay, a 60-second delay, and 120-second delay. That is the delay between the sample and the choice, okay? Or they give list length. So they show 3 samples in a row, and then they give the test. So it's kind of both increasing the delay and increasing the memory load at the same time. It makes it a little bit harder. So a list length of 3, 5, and 10. You can see how well the control animals do on this task. There are above 95% for all of these delay intervals as well as the list length. Mishkin did 3 different types of lesions in 3 groups of monkeys. The first group of monkey was a monkey that was prepared to have a lesion identical to patient HM. He called that the AH lesion, the amygdala hippocampal lesion. He went in and he did a very similar approach and a very similar lesion as the one that Scoville described. And look what we see in terms of the performance of this group. Terrible performance on the shortest delay interval, lower than 70% correct, whereas controls are up at close to 100% correct, and then it gets even worse at 60, 120, and the list lengths. They're doing close to chance, only 55% correct when chance is 50% correct. Devastating lesion, and this was a fantastic finding. I should tell you the background here, because this study is credited and heralded as the first time they were able to show a significant HM-like memory impairment in an animal model system. And you're probably sitting there thinking, so what? Of course, that's what I would predict. You already told me that HM had a lesion. But I'm skipping. This is 1978. HM's paper came out in 1957. What happened between 1957 and 1978? How come it took them so long if this was such a simple thing? It took them so long because other people, they did do hippocampal lesions or hippocampal AH-type lesions, but they did not use the right kind of memory task. Part of this work was done before Brenda Milner realized that HM could perform different kinds of performance tasks. And in fact, the animal lesion people tended to choose those kinds of tasks to test. And they said, oh my God, the animals, the monkeys can perform these tasks just fine. Maybe this isn't a very good animal model system. And they also did these lesions in rodents. And again, did not choose the right type of memory task. It took, I don't want to give you the impression that this understanding of all these tasks that are impaired or spared happened immediately after HM was discovered. It took another 20 years of testing of patient HM, 10 to 20 years, let's see, 50, yeah, close to 20 years, to really get appreciation of this. And this is what inspired Mishkin to be very careful in his task choice and really make sure he chose a task that HM himself could not perform. He was already doing it in monkeys, so monkeys could tend to perform these tasks more like humans. And so we got this severe memory impairment. But what he wanted to ask in this study, because he was able to do these more specific lesions, is here we have a damage to both the amygdala and hippocampus, just like HM. What was causing the impairment? Hippocampal damage or amygdala damage? And so he separated them out. He did one group that had amygdala damage only, and in a second lesion group that had more selective hippocampal damage. Okay? I should say that the caveat here is that all three of these groups had damage to the cortex surrounding the hippocampus and the amygdala. So you know that the hippocampus and amygdala are below these cortical areas. It's below. Do you remember dissecting out the hippocampus in your sheep brains? There's all the cortex that surround it. So to get to these cortices and the amygdala, you actually have to damage the surrounding cortex. And that was what was happening in all of these three cases. But nevertheless, it was a precise attempt to look at this. And his conclusion was the following. One, we have now identified a very viable animal model system where when you do an anatomical lesion that's similar to HM, you get what looks to be as severe a memory impairment. That's very good. Now, when I go and figure out which one of these structures is more important, I get each one alone very little impairment. Only damage together, the full lesion, causes the impairment. So he concluded that the reason that HM was so impaired is that he has combined damage to the amygdala and the hippocampus. Both of these structures must be contributing to the severe anterograde amnesia seen in patient HM. Yes? So we're going to get to that, okay? So that's, but remember, this is way back in 1978, trying early days to figure out what's going on. Clear conclusion. So, you know, from this data, does everybody see why he concluded that? Is that clear? Okay, good. So hippocampus is not doing it alone, not causing a big impairment. Amygdala not causing a big impairment, only combined amygdala hippocampal damage, okay? So let's review. Lashley, no memory structures at all. HM, Scoville-Milner, medial temporal lobe is critical. Now, what Mishkin added to this is that what's critical in the medial temporal lobe is the combined damage to amygdala and hippocampus. That's the only way you get the severe memory impairment. But you bring up a very good point, and that is there are all these pesky surrounding cortical areas. Not sure exactly what they're doing. Let me remind you that we are down into the temporal lobe. And back in the 50s to the 70s, it was called the silent lobe. We really didn't know what was going on back here. We know much more about the primary visual cortex, primary somatosensory cortex, primary motor cortex. We can follow those pathways into and out of those primary sensory and motor areas. These are much more mysterious areas. Not sure how they're organized, and very little idea of what brain areas are related to these areas. Still just trying to focus first on amygdala and the hippocampus. Okay, so let me go to this picture. This is a picture of a ventral view of a monkey brain, okay? So it's like looking at the monkey brain exactly like this. Here is the human, here is it's not a rhesus monkey that we use typically, it's a fascicularis monkey, same kind of size monkey, slightly different species. Looking very similar, all the main parts are here. Here is the cerebellum, here's the brainstem, here is the optic chiasm, here is some of these beautiful cranial nerves over here, here's your olfactory bulb, here's the temporal lobe. Okay, same thing you can see on the human, except for there are more gyri in the human compared to the monkey brain, but all the structures are very similar. So what I've drawn on the surface, let me take that away for a second. This is the cortical surface. Here are some of the brain cortical areas that are surrounding this medial temporal lobe area. I'm jumping ahead again because we didn't know what they were, but now we know that they're made up of these cortical areas. They're called the entorhinal cortex, perirhinal cortex, parahippocampal cortex, and they surround the hippocampus right here, which is just deep to this cortical area right here, and then the amygdala is right here. So really perirhinal, PR, and entorhinal, ER, surround the amygdala, and posterior perirhinal, posterior entorhinal, and parahippocampal surround the hippocampus. You have these cortical areas that surround the hippocampus and the amygdala. So what the next kind of, not next generation, but the next set of explorers in this area said is, what if I try and do a selective damage to the amygdala alone? And what if I try and selectively damage the hippocampus alone, and then compare that to damage of the hippocampus with the surrounding cortical structures, okay? So what they were able to do with amygdala, which is not shown here, but it's just deep in an almond-shaped structure right in the front tip of the temporal lobe, is they went in with a radiofrequency probe, put the probe right in the middle of the amygdala, turned it on, and basically did a selective lesion right at the tip of that probe that could damage much of the amygdala in just 1 or 2 different penetrations. When they did that, and they went back and looked at the effects of the lesion with just amygdala damage alone, you got completely normal performance. Damage to the amygdala alone is not doing any damage, even if you don't damage the surrounding cortex. But we found that if you have hippocampal lesion alone, or hippocampus together with all of the surrounding cortex around here, that's when you get the most memory impairment with what they called an H++ group. Hippocampus plus all the surrounding cortex, okay? Control memory is good performance on memory is up here, and you can see the worst performance is if you damage the hippocampus plus all the rest of the cortex that we think was damaged in patient HM. If you do the hippocampus damage with just a little bit of this cortex, you get a little bit of less impairment, but still significant impairment. Lower scores means higher memory impairment. And hippocampal lesion alone does produce a memory impairment, but it's pretty mild, okay? And so this suggested that the amygdala, shown here, when damaged alone, is not really contributing to the kind of declarative memory impairment shown in patient HM. It really seems to be the hippocampus plus all the surrounding cortical stuff, okay? I should also say that we had a hint that the hippocampus was was particularly important. And after this finding, this key hint was very valuable. The hint was that we had access to another patient, not HM, but this patient's initials were RB, patient RB. Patient RB had open heart surgery and had anoxic ischemic damage. Remember I told you those hippocampal cells are very, very sensitive? So if you have loss of blood and oxygen to your brain, you tend to get damage to these hippocampal cells. So patient RB had open heart surgery, had an anoxic event during the surgery, came out. He had what we call a clinically significant memory impairment. His memory impairment clearly was impaired compared to before surgery, and he actually was not able to work because his memory impairment was that affected. Was he as severe as patient HM? Absolutely not. But it was clear, and very, very detailed neuropsychological testing was done on this patient, RB. Unclear what was going on. Patient RB passed away, and he donated his brain to science. And they did a very, very careful histological examination of patient RB's brain. What they found was one subfield of the hippocampus, all the cells were dead on both sides. Not the whole hippocampus, one subfield. So he had bilateral damage specifically to cells in the hippocampus, and that caused a clear memory impairment that was not nearly as severe as patient HM. Now look again at these findings. These are findings from monkeys with different lesions. Again, small but significant impairment on this delayed non-matching sample task with the selective hippocampal lesion that's like patient RB. But what we get, the real impairment is when you get the hippocampus together with the surrounding cortex that's damaged, okay? So that seems to be the key structures that are important that when damaged cause severe anterograde amnesia, severe declarative memory impairment, okay? You might, the other point that I want to make is they also did a very important experiment. So to this H++ group that was showing a very severe impairment, they said, we just want to go back and make sure that this conclusion that you need this kind of combined damage is not true, just making sure. And so to this group that had hippocampal damage plus the surrounding cortex damage, a lot of surrounding cortex damage, H++, lots of cortex damage, what they did is they went in and made that very selective amygdala lesion that did not damage the cortexes at all, but just damaged the amygdala alone. And they asked whether adding an amygdala lesion to this H++ lesion made the deficit even worse. And the answer was no. You can't see it here, but there was some room to get worse here. That experiment proved that the amygdala was not contributing somehow in an additive way to the impairment. Okay, so any questions about that? Okay, now what I want to do is talk about what the heck these cortical areas are doing. Okay, why would they be so important for causing the memory impairment? Anybody have any ideas? Why would those areas be important? Yes? Parahippocampal place area is important for place recognition. That parahippocampal place area tends to be more posterior than these areas that were damaged, that are damaged here. Still parahippocampal, but very, very far back. But possibly related. It's actually a different area, but good try. Any other thoughts about what these areas might be doing? They're right next door to the hippocampus. Any idea? Why would you get a more severe impairment with kind of a grouping of areas versus just one area alone? Any thoughts? Yes. Make it more difficult for that part of the brain to communicate with the rest of the brain. That's actually an excellent answer, and that is the answer. So the answer to why we finally figured out why these surrounding cortical areas were so important had to do with the connectivity of these areas, how these areas were hooked up with the rest of the brain and how they actually communicate with the rest of the brain, okay? So remember, here are these key areas. These three cortical areas, entorhinal, perirhinal, parahippocampal areas, are the key cortical areas, together with the hippocampus, that are causing the severe anterograde declarative memory impairment, both in monkeys on delayed non-matching sample, but also in patient HM. And here is the connectivity of these areas. Now, I should say that early on, we knew that one of these cortical areas, the entorhinal cortex, shown right here, we already knew that it had very strong connections to the hippocampus. This whole box here is the hippocampus. But what we realized later is that some of these surrounding cortical areas, perirhinal and parahippocampal in particular, provided nearly 2/3 of all the cortical input to the entorhinal cortex. So all of the information that the entorhinal cortex gets that then funnels into the hippocampus, they're coming exactly from these surrounding areas. 2/3 of all the input to the entorhinal cortex comes from perirhinal and parahippocampal. Okay? So then you say, okay, well, that kind of makes sense, but then what do these areas, what kind of connections do these areas have? And are there other direct connections to the entorhinal cortex? Well, the answer is the vast majority of all the cortical inputs to the entorhinal come to these areas. But when you ask the question, what connects to the perirhinal, PR, and parahippocampal area, PH, what you see is a huge convergence of projections from lots of different brain areas, from the frontal lobe, from the temporal lobe, visual areas in the temporal lobe, including those posterior parahippocampal areas, are projecting into the more anterior parahippocampal and perirhinal areas. Insular cortex, this area is involved in somatosensory processing. Cingulate gyrus, important in decision making. Parietal lobe, important for spatial functions. So basically all sensory modalities project down and converge onto the perirhinal and parahippocampal cortices, and these areas then process information, project to the entorhinal cortex that then goes into the hippocampus. So are these just relay areas, or are they actually doing something for memory? And a lot of the work over the last 20 or 30 years have shown particularly how important the perirhinal cortex is for processing certain kinds of memory, new associative memory. You don't have to know the details there, but just know that these areas are not just relay stations. These areas, together with the hippocampus, are participating in different forms of plasticity that underlie both the fact and event memory that we know is dependent on these brain structures. Okay. So let me just end by summarizing how declarative long-term memory is stored in the brain, and just going over again this theory of consolidation, this idea of consolidation, and what is happening to new declarative memories. So we're going to talk about memory of this lecture. So this was lecture number 2 of Learning and Memory. Went over in greater detail all the different subdivisions. This includes both factual information as well as event information. Information about event information, like you might have thought that that clip from The Office was really funny. Okay, so remembering that episode is part of an episodic memory, but you also might remember what anterograde amnesia, the definition of anterograde amnesia, that is a fact. Okay, you need your medial temporal lobe to remember both those episodic qualities of today's lecture as well as the semantic qualities, the semantic information that you got from today's lecture. So if you have your hippocampus today, you'll be able to remember it. If you have hippocampal damage today, you can still understand me, but you'll have no memory of either those facts or events. If you get your hippocampal damage today, you certainly remember all your childhood. You remember all your elementary school, middle school, just as well as everybody else. Why? Because those memories are now independent of your medial temporal lobe. They're not stored there. They're someplace else, presumably in the cortex. That is spared long-term retrograde memory. But consolidation theory says that you not only need your medial temporal lobe to make any new memories, but you're using your medial temporal lobe to strengthen the memories that you made yesterday, over the weekend, over the last several weeks, maybe over the whole semester here. You are still consolidating those memories using your hippocampus So today's hippocampal damage will not only not allow you to form new memories, but will impair memories for all those categories of memories that are still being consolidated by your hippocampus, okay? So the idea is these memories are processed by the hippocampus and the related brain structures, and they continue to be processed until they are consolidated. And again, by process, I mean this is over several months or even over years of the memories being vivified, played by your medial temporal lobe, but eventually they become independent of the medial temporal lobe. They live someplace in your cortex, and so damage to your medial temporal lobe will not affect those memories, okay? Any questions about consolidation theory? And you need to know the definitions of anterograde amnesia, retrograde amnesia, anterograde memory, retrograde memory. You need to be able to describe consolidation theory. Now, I have one last question for you in the last 5 minutes. We're going back to Lashley, who was so sure and who was so influential in the 1930s, showing the law of mass action. The severity of the memory impairment for maze performance correlated with the size of the cortical area removed, not with the specific location. Why did he conclude this, and why was he wrong? Critique this experiment now that you know everything you know about memory performance. And I should also say that this maze performance was an excellent test of declarative memory, dependent on an excellent test of this form of long-term memory that is impaired in patient HM. So why was he wrong? Why did he include that? Yes? That's true. He said there's not one form of memory. There's lots of different memories located in different parts of the cortex. But he was testing one form of memory here, right? So why was he so wrong about this form of memory? Yes? Right. So she said that when you damage more, you do get more impairment. Do you have something to add to that? No, this is, so you have to start with the knowledge. I'm just going to tell you that this is a good kind of declarative memory task. And you're right that within the medial temporal lobe, more damage causes more impairment. But what did I tell you? Where was he damaging? What was his lesion? Where was this lesion? In the cortex. Throughout the cortex. If this was a rat brain, he made lesions all throughout the cortex. What's wrong with that? Yes? He never got to the hippocampus. He was lesioning the wrong part of the brain for this particular task, okay? He was actually damaging the long-term memory area, but he didn't really have the establishment of a long-term memory. But the main point is that he got the wrong answer because he had the wrong structure, okay? If he had focused on the hippocampus and said, okay, I'm going to compare the hippocampus to this area of parietal cortex, he would have said, oh my God, I get the severe memory impairment with a hippocampal lesion, and it's devastating, but I get very little impairment from a parietal cortex lesion. He didn't have that understanding. He was just trying to say, here is a region. I think it's important for memory. I'm just going to damage different parts and see what I get. And he got this law of mass action. And this was true for his data, but it actually, you can understand, it does not define our current understanding of how memory works. Is that clear? Is that clear why that's not, why he made a mistake? And this is a really important example. Why? Because we're all making that mistake. We're making that mistake now in neuroscience, there's somebody who has made this mistake because they didn't know. They were exploring one thing. They've made some conclusion, and it seems right for the experiment that was done, but we will find out in the future that that was the wrong experiment. Here is something we can look back, and now we know why the experiment was wrong. You need to understand that we're going to be asking you that on the final. Okay, see you on Wednesday.